Hi, we're going to do reproduction. This is chapter 27. So we want to review the process and the outcome of meiosis. Remember that when we have reproduction, we can do mitosis. And in mitosis, we're going to get two identical daughter cells, or we can do meiosis. And that's when we're going to form sex gametes. And the sex gametes are going to be the eggs or the sperm as far as humans go. We're going to describe the development, the structures, and the function of the male reproductive system, and then identify the factors that can contribute to male infertility. Then we're going to describe the structures and functions of the female reproductive system, including correlating the hormonal and structural changes during the menstrual cycle, and identify any factors that contribute to female infertility. Okay, so here we go. So let's just review really quickly mitosis versus meiosis. So this side is going to be mitosis. And if you have any Play-Doh handy or even just pieces of paper, it's really a good idea to sort of review this hands-on. So you can cut out some paper or you can make these out of Play-Doh. But the first step of both mitosis and meiosis is always going to be chromosome duplication. Okay, and so right here, this is our parent cell. Our parent cell, we always say it's 2N. 2N means that there's going to be 4 chromatids here. Okay, and when we have that, 2N means that we are diploid, D-I-P-L-O-I-D. Okay, or we can just be N, and we're going to be haploid. Haploid means half, technically. Okay, and so we're going to go over mitosis first, and in mitosis, we're going to develop 2 identical daughter cells. Okay, so remember that parent cell is going to start in as a diploid cell. We're going to do chromosome duplication, and so that's when we get to the first phase of mitosis. Now we can abbreviate mitosis as P-MAT. That's the letter P, letter M, letter A, letter T, and that helps us to remember the stages or the phases in order. We have prophase, metaphase, anaphase, and telophase. Okay, so during prophase, what happens is that we have these duplicated chromosomes. So we have what are called 2 sister chromatids right here. Okay, then at metaphase, we're going to have those chromosomes align at the middle. That's what meta means, in the middle. It's sort of at an equator sense, and it's because we have these extensions coming out right here. Okay, and they're sort of attaching on. These are called spindle fibers. Okay, and those spindle fibers sort of attach in and help to align those chromosomes and metaphase. Then at anaphase, we're going to go into a little bit more detail here, but in anaphase, this is where the chromosomes actually, or the 2 sister chromatids, are going to start to pull apart from one another. Okay, and then during telophase, we get sort of a peanut-shaped cell because we're going to start to see the new formation of the nuclear envelope that's going to surround these chromosomes, as well as the sort of the division. So we're going to start to see the cell membrane sort of shrink in so that it can divide into 2 cells. Okay, so then we have these daughter cells right here. So this is that parent cell again. Remember, it's 2N or diploid, and now these daughter cells are also going to be 2N or diploid, so identical cells, right? Now, during meiosis, we want to get haploid cells, and the reason we want to get haploid cells is because we do this during like during sexual reproduction, which means that we need to have sperm from the males and an egg from a female in order to really get the full DNA for a human. Okay, so this is meiosis. Okay, so remember we started with that parent cell, so chromosome duplication is going to occur. That's always that first step. Okay, and then we have 2 phases of meiosis. We have what is called meiosis 1 and meiosis 2. Okay, now meiosis 1 is very similar to mitosis. Okay, so we have prophase. We're going to line up what are called homologous chromosomes. Then we're going to line up along the middle during phase. Okay, now what we do have is when we sort of line up these homologous chromosomes is that we can have what's called crossing over, and that's where a little section of the DNA can sort of cross from one side to the other. So maybe it's the part with the hair color or the eye color, right? And so in my family, for instance, my brother has blue eyes, I have green eyes, my parents have brown eyes and hazel eyes. And so there's some crossing over that can occur so that we can get genetic diversity, okay? So crossing over is going to happen here in meiosis 1, okay? Then we have that metaphase happening, and then we get anaphase and telophase. Now at this point, we're going to sort of separate, but the sister chromatids are going to remain together here, okay? Unlike mitosis, where they separate. So now we have haploid cells, okay? And once that happens, we're going to continue to we don't do any further chromosomal duplication. Now the sister chromatids can divide even further, and we get daughter cells of meiosis 2. They're going to be considered haploid, right? So you can see here these diploid, right? We are 2N, we have 2 sets here. Haploid. We only have 1 set, so that means that each cell has 1 gene for eye color, 1 gene for hair color, okay? Whereas these have both genes, right? So remember that with humans, for instance, we get a gene for hair color from our mom, we get a gene for hair color from our dad. Okay? All right, so this is just showing you meiosis in a little bit more detail, right? So this is prophase 1. Now remember that duplication has already happened. Okay, and so during prophase 1, we get the chromosomes actually we're going to condense, and we also have a nuclear envelope. Remember that nuclear envelope is surrounding the nucleus and protecting it, okay? But in order for crossing and for us to divide into 2 separate cells or even 4 separate cells eventually, we have to have that nuclear envelope degrade, okay? So the nuclear envelope will break down and we'll do what's called crossing over at that point. Remember, that's when those homologous chromosomes can cross over, meaning that the brown eyes from one parent can cross with the blue eyes from the other parent. So again, get genetic diversity. Okay. Then we have metaphase 1. Okay. So we can see those chromosomes are going to move to the middle or the equator of the cell. Then we have anaphase. This is where those homologous chromosomes are going to move towards opposite poles of the cell. Okay. And then we have telophase, and telophase is that peanut-shaped cell, right? So we're going to start to see the development of a nuclear envelope if this was mitosis. But because it's meiosis, we're not going to see that nuclear envelope form until over here at the end of meiosis 2. Okay? So these chromosomes are going to gather at the poles of the cells, and the cytoplasm is going to start to divide. We call that line a cleavage line. And then cytokinesis is when we have those 2 separate cells. Okay. Now we're going to start meiosis 2, and so we go through those same phases again. We go through prophase, metaphase, anaphase, and telophase all over again. Okay, but the way that we do this is just a little bit different. So now we have some new spindles are going to form around those chromosomes. The metaphase 2, we're going to line them up at the equator, and anaphase, they're going to start to pull away. They do that using this centromere. A centromere is lined up along the edges. That's what that is here. And that's where those spindle fibers are sort of coming out of. Okay. They move to the opposite poles of the cells here at anaphase, and then telophase is that we get that sort of peanut-shaped look again. We start to form that nuclear envelope around each one of those chromosomes, and the cytoplasms can divide. And then cytokinesis give us those 4 separate cells now, right? And now we have haploid cells because we did not duplicate again. Okay? Now again, I remind you, this is really helpful if you use like Play-Doh or something like that to help you with the formation of this. So let's start with that male reproductive system. So the male reproductive system, we have the penis, the scrotum, the testes, the epididymis, the vas deferens, the seminal vesicle, prostate, the bulbourethral gland is also called the Cowper's gland, and the penile urethra. Okay? And it's really important that you get familiar with saying these terms, especially if you're going into nursing, and that you're fully aware of where they're located within the male body. Okay? And so right here, this is going to be the shaft of the penis. And within the shaft of the penis, you're going to see the urethra. Okay? This is the scrotum. The scrotum is the sac that the testes is going to fit into. And then the epididymis actually surrounds the testes. Okay? This is the prostate gland. Okay. And right there, you're going to see that urinary bladder. Now, the urinary bladder is not part of the male reproductive system. It is part of the urinary system, okay? But it's right there together, okay? All right. So we talked about the sort of meiosis, right? And so reproductive cells are going to go through meiosis, not mitosis, okay? Now, they also do technically do mitosis, so just to be clear, they're actually doing both. So we have stem cells, right? And stem cells have 46 chromosomes. These stem cells are called spermatogonia for males, okay? And then we have the sex gametes. Sex gametes are going to have 23 chromosomes because 23 chromosomes is half what a person or a human should have, right? We should have 46 chromosomes. Each one of your cells has 46 chromosomes. The germ cells are going to undergo meiosis to produce those sex gametes, right? So here's that spermatogonium for one, spermatogonia for more than one, okay? And they go through mitosis, meaning that they're going to be diploid and they're going to end at diploid. And some of them are going to remain spermatogonium so that they can go ahead and still remain sperm cells, okay? So that they can do more reproduction. And then we can have this primary spermatocyte. The primary spermatocyte is going to go through meiosis 1 and it forms a secondary spermatocyte. And then we're going to go through meiosis 2 to form what are called spermatids. Now, spermatids have to undergo what's called spermiogenesis. And that's where they become mature sperm. Now, this is what a cross-section of the seminiferous tubule. So this is the testes, and around the testes is the epididymis. And then we have all these seminiferous tubules right here, okay? And this is where sperm maturity occurs, okay? And so out on the edge is where we're gonna have those early spermatids, the spermatogoniums are gonna be out here. And then as we move towards the center of the lumen, and the lumen, remember, is that opening, is where we're gonna have the more mature sperm. So the prepuce is actually the foreskin, and that's found on uncircumcised males. Once circumcision has happened, then they've just removed that prepuce or the foreskin. There's a higher incidence of UTI or urinary tract infections and penile cancer, and there can even be a greater infectivity with HIV in uncircumcised males. The scrotum is a muscular sac, and that's going to hold the testes. The testes actually descend down into the scrotum. They do that during the 7th month of gestation. The reason that they need to do that is because they have to be kept at a lower body temperature. So if an undescended testicle, meaning that it does not descend down when it's at about that 7th month of gestation, then those testicles or testes can have a higher risk of cancer. That's because they've been exposed to a higher body temperature, right? There's a muscle in there, it's called the cremaster muscle. The cremaster muscles are going to help to pull the testes up closer to the body when it's cold outside, and then they relax down when it is warmer. Okay. This is testes in the epididymis, right? So here's the head of the epididymis. You can see it right here wrapping around the testes. Okay, this is the testes. Within the testes, we're going to have those seminiferous tubules. Remember, the seminiferous tubules is where we're going to have the sperm production. The sperm are going to mature within the head of the epididymis, and then they're going to leave out through what's called the ductus deferens. Okay, that's the tube for the mature sperm to leave testes during ejaculation. Okay. This is showing you the descent of the testes. So what happens is, this is 2 months. This is in utero. Okay, so this is the testes are going to remain the same size while the rest of the fetus is going to grow. So the relative position of the testes is going to start to shift, okay? And they're going to slowly start to descend down. So the testes themselves are made of lobules, and those lobules contain those seminiferous tubules inside of them. The seminiferous tubules are going to contain the spermatogonia. Remember, spermatogonia are stem cells, and they have 46 chromosomes. The spermatocytes are undergoing meiosis. Remember, they've completed meiosis 1. The spermatozoa are the sex gametes. They have 23 chromosomes. And then the Sertoli cells are actually supportive cells. They surround the spermatocytes. Then in between the seminiferous tubules, you're going to see the interstitial cells. Interstitial cells are also called Leydig cells. and those are going to secrete the testosterone. They do that when we've released luteinizing hormone, or LH. LH is going to promote spermatogenesis. You should hopefully remember that from our endocrine section. Those Sertoli cells are going to secrete what is called androgen binding hormone, or HBP. That's going to help promote or bind the testosterone to stimulate spermatogenesis, and they extend physically around the sperm cells themselves. They produce what's called a blood testes barrier, okay? And remember that these sperm cells, they only have 23 chromosomes. 23 chromosomes is not a normal amount of chromosomes for your body cells to have. Therefore, your immune system would actually attack them, okay? And so this blood testes barrier helps to keep those immune cells from attacking those sperm cells, and it keeps any surface antigens from developing on the sperm cells that are going to enter into that bloodstream. Spermatogenesis occurs in response to follicle stimulating hormone, that's FSH. It progresses from the perimeter of the tubule towards the lumen. So remember, we have the mitosis of the spermatogonium along the outside. Then we have this primary spermatocyte. They've completed meiosis 1. We have the haploid spermatids, and then spermiogenesis is the mature, they're going to mature to sperm. This is just sort of showing you that whole idea of spermatogenesis. Okay. You can feel free to read over that as you'd like. Okay. But this is sort of our key here. So we've already gone over this. This is that spermatogonium. This whole process is called spermatogenesis. That is not the same as spermiogenesis. Okay? Spermatogenesis is the whole process. Spermiogenesis is maturing those spermatids to the spermatozoa, or the regular sperm that you would think of. Okay? So this is what a mature sperm actually looks like. It has what's called an acrosome. The acrosome is along the edge, or the head, and it contains enzymes. Those enzymes are going to penetrate the egg during fertilization. The nucleus contains 23 chromosomes. That's important, right, because that's haploid. The midpiece has the mitochondria, and the mitochondria are going to help to make the ATP so that we can have the tail movement here. The flagellum on one, or the flagella for a bunch of them, is sort of going to make this whip-like movement, and allows it to swim towards its target. This is capacitation. So spermatozoa has to undergo capacitation so that it can be fully functional and motile. So first, the spermatozoa is going to become motile when it's mixed with the secretions of the seminal glands. Then they become capable of the successful fertilization when they're exposed to the conditions of the female reproductive tract. Remember that fertilization does not happen in the uterus, okay? We're going to talk about that in just a little bit. But fertilization happens actually in the fallopian tubes. So the epididymis is going to secrete a substance. We don't know what that is yet, and that's going to prevent premature capacitation. The epididymis is going to cover over the apex of the testes. They have some nurse cells in there. They're going to feed the sperm. These nurse cells are really helpful to sort of take care of those sperm. Okay. And the epididymis has 3 functions. It monitors and adjusts the composition of the fluid that's produced by those seminiferous tubules. It can also act as a recycling center for any damaged spermatozoa, and it stores and protects the spermatozoa and facilitates their functional maturation. This is the site where spermatozoa learn to swim as they fully mature. The vas deferens is, maybe you've heard of a vasectomy. The vasectomy means that they're going to clip off this tube. So here's the vas deferens right here. Okay, so you can see here's the testes, there's the epididymis, right? We're going to be maturing our sperm. And then this right here is the ductus, or the vas deferens, right? And it's coming all the way along. Okay. Okay, so the vas deferens can actually be accessed very easily for the vasectomy because it's right up here near the bladder. It's going to enter the pelvis via the inguinal canal as part of the spermatic cord. Remember that spermatic cord is going to contain the gonadal artery and the vein. And the vas deferens is going to loop over the bladder from front to back. Semen is going to consist of sperm and the fluid that's secreted by the seminal vesicles. Here's the seminal vesicle right there. Okay. And there is that prostate gland right here. Okay, so look, it's going to travel. The sperm have to travel from this epididymis through the vas deferens. It's going to then be mixed in with the seminal vesicle fluid, and then it's going to enter into the urethra to be ejaculated. So that sperm has to be mixed in with that fluid from the seminal vesicles, the prostate, and the bulbourethral gland before it's considered official semen. Okay. The seminal vesicle is going to sit posterior or behind the bladder, and it contributes about 60% of the seminal fluid. It also has what's large amounts of fructose, and that fructose is used by the sperm to fuel the mitochondria so that it can generate the ATP with the energy for the flagella to be able to do that whip-like motion. And then the prostate sits anterior to the rectum. At the base of the bladder, it surrounds the prostatic urethra. It secretes an alkaline fluid, which is sort of a base to, containing citrate, and it's a buffer because the vagina has lactic acid in it because it has lactobacillus bacteria in there that are producing lactic acid. So we need a buffer. The bulbourethral gland is also called the Cowper's gland. It secretes the mucus to lubricate the penile urethra during the semen passage. Sometimes it's called pre-ejaculate, but it can contain sperm. And then ejaculation is actually mediated by the sympathetic nervous system, remember, fight or flight. So what happens is that it constricts the smooth muscle in the vas deferens, and it creates that milking motion to help move the semen through, same as the peristalsis. This is some hormonal control of the testes here. The hypothalamus is going to release gonadal releasing hormone. This should be review from our endocrine system. So the gonadal releasing hormone is going to then stimulate the anterior pituitary to release FSH and LH. Here's that FSH and the LH release. Okay. So the LH, or the luteinizing hormone, is going to act on those Leydig cells to release testosterone, and the FSH, or the follicle stimulating hormone, is going to release stimulate the Sertoli cells to release ABP, androgen binding protein. That ABP is going to bind to the testosterone, and that helps to keep it at a higher concentration. This is a negative feedback loop, so as we get more of this, then we're going to turn this off. Okay. We also have inhibin. Inhibin is going to negatively feedback as well to that anterior pituitary so that it can inhibit the further release of FSH, or follicle stimulating hormone. The testosterone is also going to negatively feedback. The penis has some erectile tissue. The erectile tissue allows the penis to stiffen for intercourse. So it has what are called corpora cavernosa. You can see them here and here. And then it has the corpus spongiosum. Okay. So erection is actually triggered by the parasympathetic nervous system, and that releases nitric oxide, which is going to cause the relaxation of the arterial smooth muscle, and then it engorges with blood in the erectile tissue. So here is flaccid penis, and here is an erect penis, and so you can see the blood flow change. In semen, there's 20 to 100 million spermatozoa per milliliter of semen. Seminal fluid is, again, that mixture of the secretions from the seminal gland, the prostate gland, the nerve cells, the epididymis, and the bulbourethral glands. There's also enzymes in there. So we have protease, and protease helps to dissolve the mucus within the vagina. We have seminal plasmin that helps to kill bacteria, including any E. coli. Prostatic enzyme is going to help to coagulate semen. It does that within a few minutes of ejaculation, and it converts that fibrinogen to fibrin. However, about 15 to 30 minutes later, we have fibrinolysin, which is going to liquefy that clotted semen. These are some hormonal changes that you can see at puberty in boys, right? We've already sort of talked about this whole process of what's happening as far as the sperm production and the stimulation of growth and the development of male sex characteristics. We talked about this when we went through the endocrine system. I would encourage you to sort of go over this chart, and you can see that the blue is inhibiting and the red is stimulating. So there's different causes of male infertility, specifically particular surgeries, like if you've had a vasectomy, okay, or prostate surgery. Any damage to nerves can cause some male infertility. So we can have problems with producing the hormones, so the hormonal control. That would mean that there may be an issue with the hypothalamus or the anterior pituitary, right? We can have some blockage of the sperm transport. So an infection could cause that. A vasectomy obviously could cause that. An absence of a vas deferens wholly can cause that. There can be sperm production causes, so failure of the testes to descend, any infections, torsions, which is a twist motion, varicella, which is inflammation of the veins, drugs, radiation damage, sperm antibodies, and even excessive heat. And then ejaculation and erection problems can be caused by damage to nerves, timing of intercourse, and even medication. Okay. So for review, I would like you to please describe the development, the structures, and the function of the male reproductive system. Make sure that you can do this in great detail, that you can really talk about what those different male structures are, where they're located, what the development is like for the males regarding the descent of the testes, and what's the function of the male reproductive system, and then any factors that may contribute to male infertility. And we will pick up next time with female reproduction.